Welcome to Cerba, Asia. You are about to begin a tour of our Center of Excellence in Immuno-Oncology in Taipei. Please feel free to look around to fully enjoy the experience. Let's start. Specializing in immuno-oncology, our lab gathers specialty testing methods for clinical trials under one roof, bringing medical and technical expertise to manage and analyze samples safely and efficiently. Here is the sample reception room. Every day, we receive and store a variety of biological samples. Each sample is barcoded and tracked through its testing journey in our laboratory with our clinical trial management system. After registration in our system, samples are carefully stored in the suitable storage units, which are connected to a temperature monitoring system that sends alerts if needed, ensuring compliance with storage requirements. A liquid nitrogen storage tank can preserve clinical samples at -190 degrees Celsius. To guarantee sample conservation in its testing environment, the entire lab is temperature controlled and humidity monitored. Flow cytometry measures immune characteristics at a cellular level to assess patient response to therapy and to predict patient-specific treatment outcomes. Flow cytometry uses single-cell suspensions from a multitude of biological matrices such as whole blood, bone marrow, fine needle aspirate, and other soft tissues or bodily fluids. We are equipped with all necessary laboratory equipment, including centrifuges, microscopes, biological safety cabinets, and gentle max dissociator to mechanically separate tissue into a single-cell suspension for downstream analyses. Our BD Fax Lyric Flow Cytometer offers 3 laser, 14 parameters, 12 colors with forward and side scatters, capabilities to analyze cells that have been stained with fluorochrome conjugated antibodies. Using this technology, Cerba Asia can develop custom assays tailored specifically to satisfy customers' needs. This technology allows us to evaluate phenotypic and functional cell characteristics, enumerate cell subsets, determine receptor binding, and monitor immune system health during the conduct of your studies. Our fully integrated clinical trial management system and our expertise in flow cytometry will help you extract critical insights for your drug programs. With pre-validated panels in a multitude of diseases and a global standardized procedure, we ensure the consistency of your assay. You are now entering the peripheral blood mononuclear cell, or PBMC laboratory, where samples that require immune cell separation are processed. As a flow cytometry support lab, it may be necessary to extract peripheral blood mononuclear cells, PBMC, from whole blood. We are then able to slow freeze PBMCs stored in vapor phase liquid nitrogen for potential retrospective analysis. Here, we are equipped with a Class II biological safety cabinet where we purify and process lymphocytes such as T cells, B cells, or NK cells. A cell culture CO2 incubator maintains the necessary environment for cell growth and activities. We have 2 different cell counters, a Luminex Mu cell analyzer and a Count S3 automated cell counter. These 2 systems utilize different but complementary methodologies to differentiate live and dead cells, which is important in any tissue culture experiment. Histopathology is the gold standard in the clinic for tissue analysis. We work with a partner laboratory to perform sample embedding and sectioning on biopsied samples before staining and analyzing them in our facility. Cerba offers a wide range of staining methods, from H&E staining for tissue architecture evaluation to immunohistochemistry for evaluating spatial context, as well as the expression levels of classic clinical diagnostic targets or novel exploratory biomarkers. In precision medicine, understanding the patient's genetic profile is critical. Next-generation sequencing is an ultra-high throughput technology developed for scalability and speed that can be used to evaluate DNA and/or RNA collected from a patient. In our laboratory, this room is where we perform DNA and RNA extraction. Once the patient's genomic material is extracted, the sample undergoes library preparation. Here, we offer library preparation for targeted gene panels, whole exome sequencing, and whole transcriptome sequencing. In this room, the library fragments are further prepared and adhered to a flow cell. The sample is now ready for next-generation sequencing. Sequencing takes place here on an Illuma or ion torrent platform. During sequencing reads are generated along with a quality control assessment. The data is now ready to be analyzed. After specimens have been analyzed using our testing capabilities, lab operators archive and store the specimens in accordance with SOPs. All data is managed and analyzed through validated digital pathology solutions. For IHC like Halo or Visiopharm and/or bioinformatics pipeline, and easy-to-understand client reports for NGS. Each workstation is connected to our global secure network with a VPN. Our on-site data center is equipped with a server and backup to avoid any data loss. The system administrators ensure the data's confidentiality, integrity, and availability. With Cerba Asia, you can benefit from the state-of-the-art services in flow cytometry, cell isolation, histopathology, and next-generation sequencing. We provide you with the information and deliverables you need to efficiently get targeted medicines to patients with unmet medical needs. Cerba Asia is committed to providing our clients with services of the highest quality. We invite you to join us in pushing the boundaries of precision medicine. Thank you for visiting our lab.